I'm Janet Rowley. I'm delighted to welcome all of you to what I'm sure will be a wonderful series of lectures this whole afternoon. And I'm especially pleased to introduce the speaker for this particular session, Robert Waterston. I was fortunate enough that Bob, when he was here as an MD PhD student, worked with Frank Fitch and my husband for his PhD thesis. And so he graduated in 1972. Therefore, you can see that I've known Robert for a long time. And in many ways and many contexts since his graduation. He was telling me today that this was the first time at the Convocation this morning that he'd been in Rockefeller Chapel since his graduation in '72. So I think it was good for him to see that the chapel is still there and to hear the organ and participate in the other events of this morning. And I'm very struck by the fact that Bob Zimmer quoted from Harper and also from the first commencement speaker that one of the major goals of the university is teaching and research, and or education and research. And I think that again, Bob sort of exemplifies that in his career, both abroad and at WashU, and now at the University of Washington. He's, I think you're in for a treat, because not only is he going to talk about the human genome, but other aspects of research that really shows you the breadth of the impact of the sequencing of the human genome on many of our activities. And I won't take more of his time, but I do want to introduce Dr. Waterston as the Gates Professor and Chairman of the Department of Genomic Sciences at the University of Washington. And he's going to talk to us today about the Human Genome Project, its impact on science and medicine. Bob.
Thanks, Janet, and thank you very much for inviting me and acting as my host. It's been my pleasure to be part of the 500th Convocation ceremonies. It's quite an occasion. I did wonder how the university started in 1509, but then I understand that convocations are held every quarter and then some. So, but it's very impressive. And I thought it would be a nice occasion in this context to look back on the human genome and its impact. This is a big project. I was lucky enough to play a central role in the early days and continuing on into the human sequencing. And it was announced, when the first draft was done, it was received great hoopla and many promises of wonderful things to come. And I think in truth, its impact on medicine so far has been very limited. That while we have this wonderful information, it helps us to understand what's going on. As I talked to my GP, and I said, well, would you like to start to get people's genotypes? He said, I don't know what I'd do with them. And so I think it has yet to come in terms of influence. But the stories I'm going to tell you about today may persuade you that the day is not so far off, where it will have significant impact. I think its impact on science has been more profound and more immediate. People can survey across organisms. They can use the genome to navigate from fruit flies to worms to humans to yeast and we can all talk to one another in a way that we couldn't imagine. And we can now look at genomes as a whole and try to understand. And the last part, I'll talk about that. And so I'm going to talk about 3 different examples. I'm going to talk about discovering genes for traits, particularly in humans. And then I'm going to talk about discovering genes driving cancer formation. And then I'm going to talk about this broader issue of, given the genome sequence, can we actually use that to have a comprehensive molecular description of an organism? And the organism I'm going to take is my favorite organism, the nematode Caenorhabditis elegans. Its name is longer than it is. This is a millimeter-long worm. Okay, so people know that have known for a very long time that many traits are hereditary. This is me after camping this summer, and my wife. And when we had children, we had the expectation that they would be some kind of a mix of the traits that we have. And that's been a long-standing sense of humans, that they know things, they've expressed it in various ways. Things run in the blood, it's in the family, whatever. People have known about that. But in the beginning of the last century, we started to know more about it. And people have studied particular traits, eye color, for instance. And people have known for a long time through breeding, domestication, dogs here. Ooh. I don't want everybody to go to sleep without me at least seeing that you're asleep. Can we have that's somewhere in between. There we go. Good. Okay. And so people, the early geneticists at the turn of the last century began to understand that some of the principles involved. They didn't know what the molecule was. They could tell that that individuals received a gene, as it was called, one from each parent, and they passed on one of those 2 genes to their offspring. So we're all a mix of our parents, but not a summation of those. But what the molecule was behind that was a total mystery. And if you go back and read papers from the 1930s and 40s, it's kind of fun to see how they struggled with what is it about the cell that has that can store the information. And it was this man, Avery, Oswald Avery, in 1944, who published the first evidence that it was actually DNA that had the genetic material. This was in bacteria. It wasn't widely accepted. It took a long time to penetrate through. But when Watson and Crick, Jim Watson is this fellow down here, and Francis Crick is the person on the right, at the ripe old age of 35 and 25. They made the discovery of the century in biology, certainly. They discovered the structure of DNA in 1953. And from that structure, it was clear how this model of DNA could contain information. And we now know today that it's got 2 strands, one wrapped around the other. It's very long, many millions of units long. But it's made up of units of just 4 different bases, A, G, C, and T. And importantly, the T from one side and the A from the other side are paired. T is always paired with A, and G is always paired with C. And so it's this linear order along the chain that contains information. And it's in every cell of every living being. So it's this sequence, and I'm going to talk about sequence. I'm going to use it both as a noun and a verb, meaning the order of the letters, and sequence as a verb, meaning to determine the order of those letters. And just like in language, when we represent that with letters, if you don't have the right order, it doesn't make sense, but letters in the right order make sense. And so it's this order of letters along the chain that stores the information. It's analogous, really, in many ways to a 4-bit computer code. And a very important feature of this is because of this central pairing, the A's with T's and the C's with G's, if you split the two strands apart, you can copy the second strand from the first. And the machinery of the cell knows that where there's an A, it should put in a T. And so you can create an exact duplicate of this one strand, and each daughter cell gets a copy. And so the information is preserved. And that copying process is extremely accurate. It makes errors on 1 in 100 million or something like that. And this is what it looks like. This is actually human sequence. It's 1/1,000,000th of the human sequence or something like that. So if you imagine 1,000,000 pages like this, you can imagine the human sequence. It's just orders of A's, G's, C's, and T's. But everywhere in the genome has its special order. So I've talked about genome already. What do I mean by genome? Basically, it's all the DNA of an organism. It's and people often talk about it as a blueprint. It's the instruction set. Is the DNA sequence for a worm the same as human? No, it's a different instruction set. Is the DNA sequence for a dog the same as a cat? No, different instruction sets. But the dog genome is shared by all dogs. It's the total blueprint. And so it's the information that allows the organism to go from an early embryo to an adult animal with all the behavior and form associated with it. And all that information that goes from here to there is encoded in that simple string of A's, G's, C's, and T's. Okay? So how big is it? I've alluded to the human already, but viruses are small, just 7,000 to 10,000 bases in polio and HIV virus. E. coli, these are bacteria. Tuberculosis, these are relatively larger, 5 million. And the roundworm and fruit fly, two organisms that are widely used in experimental biology, are about 20 times that size, 100 million. But when we get to vertebrates, we get a much larger genome. 3 billion bases is what the human genome is approximately. You get 3 billion bases of sequence from your mother, 3 billion from your father. You have 2 copies. It's not always more complex means bigger. The opossum has 4 billion bases. The toad has 7 billion bases. And who knows why? Okay. So, and then once, you have to realize when Watson and Crick discovered the structure of DNA, they couldn't obtain the sequence. They could infer what the structure was, but they had no way of actually understanding no method to read the sequence. And that didn't happen until the 70s, when these two gentlemen, Fred Sanger on the left and Wally Gilbert on the right, working independently, both came up with methods to chemically determine the sequence of a small DNA molecule. And on the scale of things, this was on the order of hundreds of bases. It's a long way from a genome, especially a mammalian genome. But if you take it, it was enough to get some viral sequences. And indeed, Fred Sanger sequenced several small viruses, and indeed, I think laid the foundation for the field. This was the idea that you could by sequencing, determine all the genetic information for an organism, even if it was just a virus. So with that, in the 80s, spurred on, I think, by these kinds of efforts, the wider community, spurred by Renato Del Becco in particular and others, had this idea that you could actually sequence the human genome. Now, this is using hundreds of bases at a time. 3 billion, you'd have to do, on the order of, I don't know, 20 to 50 million experiments or something like that to get the human sequence. That's a lot of graduate students. But it went forth. The idea was put forward after a lot of study, congressional committees, and so forth. The project was launched in the late 80s, and this is sort of what it felt like. Being very unprepared, climbers, minimum of sophistication, trying to take on Mount Everest. But that was the goal. Fortunately, things advanced. And so Lee Hood, shown here, changed from a radioactive sequencing methodology to using fluorescent dyes. That made it much easier. We could use a laser, detect things in real time. And so now we could do tens to hundreds of thousands of bases a day from one machine. Norm Dovicki developed methods for doing this all in small capillaries instead of big slabs, which were awkward and difficult to deal with. And ABI came out with a next generation sequencing machine that would now do millions. And with that, we had the capacity to actually generate the sequence of the human genome. The chance of doing that spurred lots of ambition and also brought in dollars. All of this had been a public effort. There was a private effort launched by ABI, headed by Craig Venter, and suddenly there was competition between the public effort and the private effort, with the private effort not clear about what it was going to do with the sequence, whether it was going to sell it to people individually or make it freely available. But both groups prevailed, and so in June 26, 2000, that's my wedding anniversary, I can remember this date, both groups announced that we had each completed a draft of the human genome. It was very highly publicized. Bill Clinton invited us to the White House. There was big press conferences. Same happened in England, Tokyo, Paris, all coordinated press conferences with lots of hyperbole. A milestone for humanity. Scientists decode the blueprint of the human body. Day for the ages dawns in medical science. Clinton hails map of the human genome. Scientists complete genetic. Human alphabet complete. Well, it wasn't quite complete. It was only about 80%, but who was counting? But eventually, we took that information and both wrote independent papers appearing within a day of each other. Science came out one day after Nature, but who's paying attention? Both had basically the same sequence because, of course, it is basically the same sequence. And in terms of this was a draft sequence. It was incomplete. The public effort continued to work on it, and 2 years later had a complete human sequence, a highly accurate sequence for everybody to use. And it was public. Nobody had to pay for it. It's in the public databases and now is indeed widely used and spurred things. Okay, so here that was 2000. We're almost 10 years later. What good is it? Well, first of all, what's in it? It's, as I said, it's 3 billion A's, G's, and C's. They're arranged in the 23 chromosomes. They're all painted in different colors and fancy ways. And as I said, each individual has 2 copies, so there's 2 copies of the yellow chromosome and so forth. That's everybody's constitution. We can detect roughly 20,000 genes in that genome. It's not an exact number. Interestingly, that's about the same number of genes that the nematode C. elegans has. So we don't have a lot more genes than the worm. So this is the next step. So we can have these genes, but what does it all mean? And how do we put it all together? And I think this is really the major challenge facing us as genome scientists and maybe as biologists today. How do you take that information that's in the genome and understand it? We're pretty good at being able to take that sequence and being able to say, well, it makes a particular protein, and we can even begin to predict the structure of that protein. The proteins are the bits that actually do the work. And we know a lot about how those proteins are organized into units, different structures in the cell, how the cell is organized. We have pretty good knowledge of that. We have a good knowledge of some of the pathways that are involved. One protein does this to a substrate, another does that, another does that, and finally you get a product that's useful. Same in regulatory pathways. We have a lot of information about that. But if you ask a biologist today to turn around and tell you from this sequence and from these proteins, how do you get a mouse? That's not clear. We're not good at that at all. And in fact, I would say that short of prior knowledge about where the genome comes from using databases and so forth, I could give anybody on the planet a genome sequence, and they couldn't tell me what organism it was going to produce. We just can't interpret the information on the genome at that level. And so if I give you a sequence over here, is it going to produce a mouse, a human, or a chimp? And we can't tell. We can't read the sequence at that level of detail. And so, to me, there are lots of different ways to approach this. Given my training and background, I'm interested in a genetic approach. And genetics has the very substantial power that you can take and not know anything about what's in between a genetic variant, so some kind of change in the DNA sequence on the left, and some kind of trait on the right. We don't have to know what's in that black box. We can establish experimentally or statistically or whatever that this genetic variant through whatever black box leads to a trait. And we can do this wonderfully in model organisms. We can do the reverse. We can start with a trait and work backwards and find out what the genetic variant is. Of course, there's our environmental influences that we have to take into account. And with experimental organisms, you can control all that. And this kind of paradigm has worked extremely well in animals like Drosophila, yeast, and C. elegans, where we can really go to town and try to work how things work. You don't have to know anything about what's in that black box. You change the sequence. It's sort of like if you pull the spark plug out of your car, what happens? If you change the sequence of the DNA in a particular place, you turn around and ask in the organism what happens. Okay, so how do we apply this in humans? Well, it's much more complex in humans. If you look around this room, or as shown in this cartoon, people are highly diverse in traits. And that's been a complication in genetics. We can't do controlled crosses. The background is very diverse. And so trying to nail things down has been a challenge. But I think in the current situation, this is actually going to be our strength. We have 6 billion people on the planet. Every base in the human genome has been changed in somebody. And we have doctors out there with shingles, hanging up their shingles, recruiting patients in, looking at phenotypes. So we can use this information, I think. And the technology is coming. And so that stems from the fact that our sequence is not the same. I've talked about the human sequence, but in fact, it's not the human sequence. Everybody's sequence is different. How similar are we? Well, we're phenotypically quite different. And by the way, that's our new Nobel Prize winner. And so there are lots of different traits. And how similar are their genomes? Well, you might say Janie has no sequence in common with anybody else, but that's not true. It's 99%, 99.9% similar. You look at Don's genome or Janet's genome and compare them, they're 99.9% identical. So 0.1% difference. But that's a lot of differences in a 3 billion base genome. That's 3 million differences between every copy. How different are we from chimp, our nearest relative? It looks amazing, the same as one of the characters above. 99%. So about 1% different in sequence. So within our species, we have about 1/10 the variation that you see between us and chimps. Can we take advantage of this variation? And so the first story I'm going to tell you about is discovering genes for traits. And this involves work from Jay Shenduri and Mike Bamshed at the University of Washington. And as a critical part of this, what's happened in the last few years, stimulated in large part by the success of the Human Genome Project, is that the Genome Project got us from hundreds to millions of bases being able to be determined to be read a day, with new technologies out there that are producing billions of bases a day. So remember, the human genome is 3 billion bases. We can begin to think about sequencing whole genomes on a very fast and cheap timescale. The lure of doing this medically on a population-wide basis, I think, probably not well-founded, but nonetheless, it's brought a lot of venture capital into this. And so we not only have one company trying to do this, but all of these companies have instruments on the market today that produce sequence at, to me, astronomically cheaper, faster, better. They took, I mean, basically, if we go back, we built a lab in St. Louis with 250 people, robots, and machines. This one instrument that sits about this big does all of that in the box. It's much easier to run one instrument than it is to guide 250 employees. So, and there are other technologies that are coming on the market, maybe next year, that are even going to be faster and cheaper, working by still different principles. And indeed, in last week's paper, IBM has entered the fray with its own effort. So I think sequencing is already fast and cheap. It's only going to get faster and cheaper. So can we apply this to humans? And Jay and Mike Bamshed have applied this to a simple inherited disorder. They have a family in which the disease is propagating. So these individuals in the blue circles have the trait, and I won't go into details. Those are different generations. But it's the simplest case. You have one gene causing this trait, and we know this because of the inheritance pattern. And so we can just do this simple arrangement. Let's see what the sequence change is. Let's look at what the trait is, and can we go backwards? And there are actually thousands of such traits out there that remain to be solved, where there's just a single gene that has, when it's changed, we know it has an observable effect on the individual. This is the gene that they went after, or the trait. It's got a long, nice name, post-axial axial acrofacial dysostosis syndrome. We'll call it Miller syndrome, okay? And these kids have problems. They've had developmental problems with their limbs and with their craniofacial bones. They just haven't, there's been a growth problem somehow in the development. They have some hearing problems. But interestingly, they have normal cognition, no problems in the brain at all. And from its pattern of inheritance, we can infer that it takes a mutation or a change, a variant in both copies of the gene. So what Jay and Mike did was identify 4 individuals from 3 different families, so 2 were brothers, and they wanted to see if there was any DNA sequence change that was unique to these individuals that was not shared with other people in the population without the syndrome. Okay, you got the picture? We just look at people without it, and we look at people with it. And is there a gene that has changes in it that are common for the people with the trait, with the syndrome, that's not shared with anybody else? And amazingly, it worked. I wouldn't be telling you if it didn't work. But the numbers are impressive. So if you look at, and this is a complicated table, I apologize. This is real data for the scientists in the audience. So these are 2 different individuals. These are the 2 brothers. And if you look at all their genes, and you look, are there potentially deleterious mutations in any of their genes? Well, the first one has almost 2,800 genes with potentially deleterious mutations in them. His brother also has almost 2,800 mutations. If you ask what genes do they both have mutations or variants in common, we're down to 2,300. Then we add another individual and another individual, but we're still left with 1,500 genes between these 4 individuals that have potentially deleterious mutations. How can we sort that out? And the reason we have so many is this basic problem that we're 99.9% identical, but we have 0.1% difference. That's 3 million differences across the genome, and these can all be shared. So what they then did was go to the databases and actually get some more sequence from individuals without the disease. And then they asked, how many genes did individual 1A have that had a deleterious mutation that was not seen in anybody without the disease? And now we're down to 31 genes. His brother had 26. Now if we ask how many he and his brother shared, now we're down to 8. And remarkably, if you add these 2 other individuals, you're down to 1. 1 gene out of this 3 billion bases they found from this sequencing effort. And they could do this in a matter of weeks. And now it's really less than a week if they're going to go at it in a serious way. Okay, so the gene is DHODH. That's shorthand for a long name we're not even going to try to say. But if you look now, not only in those kids, but in other kids as well, they've now looked at 6 different kindreds, and all the kids, they have novel mutations in both copies of the gene. So this is pretty strong evidence that this is really the gene. This is it. What does it do? This is a key enzyme, actually, in making the elements that go into DNA and other nucleic acids. It's in this pathway. It's involved in this step. You don't have to worry about that. But it makes the stuff that goes into DNA. And so that's what Miller syndrome seems to be. And people Jay and Mike were perplexed by this. How does a problem in DNA synthesis, basically, in making the raw material for DNA, lead to these problems? And remember, I was talking about this ability to go back and forth between organisms because of the sequence. Well, it turns out there's a related mutation or related phenotype in Drosophila. This was actually first, the original mutation was isolated by Thomas Hunt Morgan at the beginning of the last century. It was called rudimentary, and it was called rudimentary because its wings didn't grow right, its legs didn't grow right, and there were some problems with oogenesis. Turns out it's in exactly the same pathway. And interestingly, if you feed flies the precursors to for DNA, they can actually have amelioration of the phenotype. They don't have such deformed wings and so forth. Whether that would ever work on people, we don't know at this point. But it actually is beginning to make sense. And the idea is simple, that cells are dividing most rapidly at the formation of the limb buds. And they're maybe removed from circulation, so they're entirely dependent upon their own ability to make new DNA as the cells are dividing. So that's a good story. We don't know if it's true, but it's a good story. So now I want to move to discovering cancer genes. Cancer, as most of you in the audience know, I think it's the second leading cause of death in the United States. Cancer is basically, I view it as a genetic disease, but it's not a genetic disease that's passed on from one generation to the next, but it's caused by new variants that happen as cells divide in your body. And every time you make a new copy of DNA, there's a finite chance of an error, a new mutation. And as you're replacing your colon cells or your blood cells or whatever, cells that are growing fast, you can get mutations that on rare occasion lead to loss of growth control. As it starts to grow faster, new mutations still arise. It grows faster and faster. This is selection in your body for a rapidly growing cell that's out of control. And importantly, each cancer is a result of independent events. The mutations that happen in one person's colon cancer are actually different events. They may end up affecting the same base, but they're different events. And now, in the next few minutes, I want to tell you a couple stories from Mark O'Mara, a former postdoc of mine, who's now heading the British Columbia Cancer Agency Genome Center there in Vancouver. And these are fun stories. So the first one is granulosa cell ovarian tumors. This is a rare ovarian tumor. Ovarian tumor is not so uncommon, but the granulosa type shown here is about 5% of all such tumors. It's been difficult to diagnose, and there's been some uncertainty about it. And so Marco took a similar approach to that of Jay. They wanted to see, are there changes in the DNA sequence of this tumor cell that are different from other ovarian cancers or other variants that are in the population as a whole? Because every time you go in and sequence somebody, there are 3 million differences from in one copy of the genome, another 3 million from the other. You've got to figure out which are the relevant ones and which are common and which are not. Remarkably, all 4 individuals showed a change in the same positions. Out of this whole 3 billion bases, every tumor had this change. In the normal position, or in the normal reference sequence, as we call it, and non-tumor cells, there's a C in this position. This is the sequence at that position. And then we show above here the sequence that they got from the tumors. And in most positions, it's a G and it matches here. It's a C, it matches here. But at this position, they got a mix of G and C, and a mix of G and C, and a mix of G and C, and almost all G in that one. The mix is because, remember, you have 2 copies. In this case, one is changed. The one is not changed. One is changed, and so forth. And in that one, apparently most of the cells have either lost the normal copy or it's changed in both copies. So that was really striking, that all of these 4 individuals had the same variation. But then they went on and tested another 89 tumors of the same type, and 86 of them had the same change. Now remember, this is happening independently. Different people, it's a different event, but in every case, they had to have this change. It's really remarkable. What is this in? It's a gene whose name is doesn't mean anything. It's FOXL2. It's a gene that's known to be important in making these granulosa cells of the ovary. It directs the growth of those cells. What the G is doing at this point, this is all new information. Just was published last summer, and so they're obviously trying to figure out what this gene is doing in these tumor. But already, it provides for definitive diagnosis. You can take an ovarian tumor. If it's got this change in it, it's a granulosa cell tumor. If it doesn't have it, it's not it. And obviously, this protein is now a target for drug development. If you can, whatever this variant is doing in these individuals, if you can now interfere with this activity, you may be able to stop the cancer. Okay? Remarkable. So now there's one other story from Marco, though, that is maybe a harbinger of the future. It's a just so story. We don't know how much of it is, we know the facts, we don't know the cause and effect, because this is a single event. So Marco, being head of the Cancer Agency Genome Center, was approached by a physician who had a patient who had a rare tumor. And because it was a rare tumor, the doctor didn't know what kind of therapy to use on it. Okay? Most tests and so forth, the drug is tried on many people with that cancer, and you work out whether it works or it doesn't work. Well, this person had a rare tumor, and it wasn't susceptible to that. So they asked Marco, would you sequence it to see if you could find out what changes might be there, and if there's a change that we can do something about? That's been seen in other cancers where there are therapies. So Marco sequenced the genes in this tumor, and what he found was, and this is a complicated pathway, these are all genes that are involved in controlling growth in tumor cells or in normal cells. And up at the top here is RET. And if RET is overactive, that stimulates this, stimulates this, stimulates this, stimulates growth. And Marco's sequencing showed that the RET gene had changes in it that made it more active than normal. It wasn't subject to the normal regulation. It was overactive, and presumably leading to this, contributing to this exaggerated growth. So they then took this individual. The person had already been on erlotinib. I think that's how you say it. It's not, I don't know, but here are CAT scans of the person's lung. And at the beginning of, they picked erlotinib because it was available, I think. And they followed the tumor progression, and it grew in this month from 22 to 27. Well, a remarkable thing about the RET gene is that there are already drugs out there that target RET. And so that's sunitinib, and they put the individual person on that drug based on the sequence. It looks like it should work, but it's pretty faint. And after a month, the tumor had actually started to regress. And that was true in another spot as well. Really quite remarkable that based on this sequence, they could direct the therapy for this tumor. They didn't know anything else about this tumor except that this was the sequence. Unfortunately, because it was already an advanced tumor, it had metastasized and so forth, you're up against the numbers game. New mutations happen, and the person had a relapse. But Marco was able to follow this up and sequence the tumor from the relapsed tumor that was no longer sensitive to the drug. And lo and behold, the changes were still there in RET. That hadn't changed. But now there were changes in genes downstream. And so the tumor had acquired new mutations that made it no longer dependent on the RET activity. It was now using these other things to drive proliferation. And so it's not a happy ending, unfortunately, but it says two things to me: that we have enough knowledge and we have to be able to start interpreting sequence in cancer, in the cancer setting, and that based on that knowledge, and we can get the sequence, we can start to think about how to change therapy. There is a huge effort being launched by the NIH, or the National Cancer Institute. They're going to spend, I think, $275 million in the next 2 years, I think, to sequence hundreds of tumors of 20 different kinds, I think. I think for the first 2 years, it's 10 different tumors. And they're going to try to get this kind of information on lots of tumors, see what the changes are that are characteristic of different tumors, and start to design more rational therapy on it. It's a just so story because this is not controlled. It could well have been that this was all going to happen anyway. There might have been regression without having that drug. We don't know. It's one case, but it's an interesting case. So now I want to move to my favorite organism in the last couple minutes here and talk about, can we achieve a molecular description? And, whoops. So C. elegans, for those of you who don't know, is a marvelous organism. It has just 959 cells as an adult. It grows from an egg to an adult in just 3 days. Each adult will produce about 300 eggs. So in a week, you can end up with 10,000 nematodes. And for a geneticist, this is a big deal. It's transparent, so we can view it under the microscope and watch everything going on within it, the whole lifespan. It has what's shown up on your upper right as the series of cell divisions that give rise to the animal. This was worked out by John Sulston, who got a Nobel Prize for this in 2002. It's got just 302 neurons. It's not very smart. 302. But we know the connections of every one of those 302 neurons. So we can study this animal with an incredibly fine level of detail. And one of the things that I thought, going to Seattle, one of the things that I thought the field really needed was to be able to understand when and where genes were being used in a much more precise manner than had been possible. So we developed the following technology. And this is going to be a pretty movie. I hope you like it. This is an egg. So this is you can barely see the outside of this is one bit of DNA. This is another. These are the nuclei of the developing embryo. And we take pictures of this. We take a through focus series. We take about 30 to 35 pictures every minute. We do this for 8 hours. We end up with about 25 to 30,000 images. And now we take these and superimpose them and run them in time lapse, and you can begin to. I can't catch it at the right time. Okay. Anyway, these are you're watching the cells divide. We've labeled the nuclei with a green fluorescent protein, which is why it's green. And it stays with the DNA, and so you can, if you just follow one of those cells, maybe that's what I should do, is just take one of these and see how it is just divided. There's another one, there it just divided. And so you're watching the worm go from 2 cells to 555. That's how many it takes to make an embryo. Now, in addition, in this worm, we put in a second fluorescent protein, and that's being made under the control of a specific gene. And so this red, which shows up as yellow where it overlaps with the green, is reflecting the presence of that particular gene's activity. So where it's green, there's no activity. Where it's red, the gene is active. And we can, and importantly, we don't have to look at this. It's fun to look at it. But we have a computer program that goes in and analyzes this, finds the nucleus at every time point, determines when it divides, and looks to see if there's red or not in that nucleus. Okay? And because every worm does exactly the same thing, every worm develops in exactly the same way, we can take the results from this one experiment. Now it's moving and we give up. We can take the movie from that one experiment and combine it with results from other ones. So here's one where the gene is going to be expressed in the gut, the intestine cells. And there it's in the first 2 cells. Those are going to divide. It's got a very simple gut. There are just 20 cells in the gut when it hatches. But you can see them lining up in the middle of the animal. These are cells that are going to become muscle. And you can see them lining up on either side. They propel the animal along in its sinusoid. These 2 are making skin cells. This one starts a little earlier than that one. And so it's beginning to cover the whole surface of the worm and the cells that are on the surface. And now it should be coming up just in that one. There they are. And so we can do this. And we've done this now for something like 150 genes. And we get really quite precise patterns. And you won't make any sense of this, but for the hardcore scientists in the audience, you'll see this is the lineage. So it starts out, these lines represent cell divisions. These represent cell lifetimes. The red ones are the ones that are expressing. The purple ones are the ones that are not expressing. In between are things that aren't quite so heavily expressing. Here's one gene that expresses in these cells, but not in those cells. Here's another one that's expressed only in these daughters, but not in these daughters. A still smaller subdivision. These are now differentially placed, and here's still a smaller subdivision. And so we can begin to think about, how does this gene relate to this gene? Does this gene have to be on before this gene knows what to do? And in the worm, you can do those kinds of experiments. You can take away the activity of this gene, and with our movies, we can now watch and see the effect on that gene. And so we're trying to accumulate. We're targeting in particular the genes that regulate other genes. These are the master regulators or whatever. There are about 300 of these that are active in the embryo. We've done about half of them, and we've got the patterns for them. And so we're starting to see interesting patterns. And the idea is that with other work that's going on in other labs, that we'll actually be able to tell the series of events that lead to these changes in gene regulation in the embryo in detail. Okay, so I've told you about 3 examples: discovering genes for traits, genes driving cancer, and the molecular description of the organism. I just want to end with a brief digression or whatever, riff on the personal genome. So with this improving technology, this ability to sequence whole genomes, a number of individuals have actually had their genome sequenced. Jim Watson, the discoverer of DNA, not only lived to see the whole human genome sequence, he lived to see his genome sequenced. Craig Venter also had his. He sequenced his own genome. This person is a Swiss entrepreneur. He paid $350,000 to have his genome sequenced. And now Steve Quake built a machine that did his. There's finally a woman, an African woman was sequenced, 2 Koreans, 1 Chinese. So more and more individuals will have their genome sequenced as it gets cheaper and cheaper. What good does it do you today? There's been a lot of money put into various studies, and so you can look at what variants are associated maybe with diabetes and hypertension and so forth. Jim Watson, however, when he got his genome sequenced, wasn't interested in what was in there. And in fact, the one place in the genome where we had strong predictive effects, we could predict whether he's going to be susceptible to Alzheimer's or not, he said, I don't want to know. And he specifically told the people who sequenced it that they could not release his sequence from that. They could not. They couldn't tell him. They couldn't tell the world. And why? Because there wasn't anything he could do about it. So we're in the position where we can assign people risk, but we can't make any much to do about it. And in fact, you know, if I sequence your genome today, I might be able to tell you had a 10% increased risk of diabetes. Would that make you change how you're living? Probably not. Maybe have a 5% increased risk of heart attack. People smoke, and that's astronomically higher risk than anything like that. And we don't get people to stop. So I think at this point, we don't know enough about the genome to have it had so that it hasn't had much of an effect on medical practice. But I think the couple stories that I told you today about the ability to associate genes with variants and phenotypes in a much more straightforward manner, in a high throughput manner. Jay has got a grant to do this Jay and Debbie Nickerson and Genome Sciences have a grant to do this, basically a Mendelian disease a month. They're going to sort out one of these every month for the next 2 years. They also have a grant to look at 10,000, the genes from 10,000 individuals who have common diseases, to look for rare variations in those people to see if those have more of an effect. They're stronger predictors. So things are going to change. And so I would say, while we're not there yet, I'm optimistic that in the next 5 or so years, things will happen. And I'm guessing that it'll actually happen in cancer first, because we know it's a costly therapy, therapeutic decisions involved in that. And compared to that, $10,000 for a genome doesn't sound like much. So with that, I'll thank you for your attention and be happy to take questions.